Okay, with that, I would like to introduce Romain Brette. He is a research director in computational and theoretical neuroscience in the Vision Institute in Paris. And his main interests are neural modeling of perception and spike initiation. And with that, I hand it over to you.
Hi, can you hear me?
Yeah.
Okay. And can you see the screen too?
Yes.
Okay, perfect. Okay, so thanks a lot for the invitation to this really interesting workshop. So what I want to present today is really an ongoing project, not at all some finished work, but it fits the theme of the workshop, so I hope you will like it. So it's about Paramecium, which, as you said, is a unicellular organism, an eukaryote, and it has been called by a number of authors a swimming neuron, and I'll explain why. And so this is ongoing work with several collaborators that are listed there. I will present them at the end. So Paramecium is a unicellular organism. It's an eukaryote, which is fairly large. So here you see Paramecium caudatum, one of the species. It's 250 micrometer long. You can see that it's covered with thousands of cilia. So it beats its cilia to swim in fresh water. So you can find Paramecium in lakes and ponds and so on, freshwater all over the world. And it has been on Earth for a long time, several hundred million years. On the right, you can see a specimen of Paramecium fossilized in amber, Triassic amber, so at the time of the dinosaurs. And so here is a trajectory of Paramecium. So Paramecium swimming on a slide, Petri glass actually, Petri dish actually. And so it swims in spirals, and you can notice sometimes that it changes direction. And this is called the avoiding reaction. So it changes direction either spontaneously or when there's a stimulus like an obstacle, so mechanical stimulus, as you can see here. And if you look a little more closely, you can see that it's not just a change in direction, it actually swims backward for a second or a few hundred milliseconds, then turns and swims backward again. And so why has it been called swimming neuron? That's because this avoiding reaction is triggered by an action potential. It has been known for a long time, since the 60s. And in fact, in the 60s and 70s and a little bit into the 80s, there was an active community working on Paramecium from a neuroscience point of view. And it was not a niche kind of line of work. It was published in like mainstream journals like Science and Nature and so on, but it's been a little bit forgotten. So Paramecium has an action potential which is calcium-based. It has calcium voltage gated channels in cilia, and so when you depolarize Paramecium, either with an electrode current or with a stimulus, then you open the ciliary channels, calcium gets into the cilia, and that triggers a change in orientation of the cilia, so the cilia beat in the other direction and the cell swims backward. Then calcium is expelled or buffered, and the cilia reorient and the cell swims forward again. So in fact, people have been interested in Paramecium for much longer time than that. In the end of the 19th century, beginning of the 20th century, there were a number of scientists interested in Paramecium behavior, so looking at Paramecium swimming with a microscope. So the most known of them is probably Herbert Jennings. He's written a really great book in 1906 called The Behavior of the Lower Organisms. So I think if you're interested in the topic of this workshop, it's a really nice book to read. So it's about several species, including Paramecium, amoeba, and some other species. So, so Jennings and others have studied the behavior of Paramecium. One of the main determinants of Paramecium behavior is the avoiding reaction. So basically the principle of behavior as described by Jennings is the following. If the cell finds itself in a situation that is not good, then the avoiding reaction is triggered, and that means that the cell swings backward, comes from where it was coming from, and then turns and tries a new direction. In fact, although it looks like a random direction, it's only pseudo-random in the sense that it always turns towards the same direction, which is away from the mouth, but because it's also spinning on its long axis all the time, well, sometimes it seems to turn left and sometimes right. So anyway, it looks like it's changing direction randomly. And with just that, you can explain, well, some more sophisticated behavior. For example, chemotaxis. So this is also, all of these are drawings extracted from the book of Jennings or some papers from the 19th century that he wrote. And in this one, he shows what happens if you put a little drop of weak acid, and he observes Paramecia gathering inside the drop. So they show chemotaxis. How does that happen? Here you can see a sample trajectory. It's just drawn, from observation directly. And the cell swims straight into the drop by chance. And then when it hits the boundary, it kind of bounces. And that's through the avoiding reaction. So basically what happens is that while it's swimming in the weak acid, it adapts to this new solution so that when it hits the boundary, there's a stimulus, there's a chemical stimulus there, and that triggers the avoiding reaction. So with just that and adaptation, you can get chemotaxis. As a side note, what's interesting compared to mammals and metazoans, usually when we think of motricity, we think of it in terms of motor control, either feedback control or planning, and all that involves some kind of measurement. But here it's a little bit different, in fact. It's more trial and error, in fact. There's no direct feedback in the sense that you know where to go or you know what error you've made. It's just something is not good, I try something new. It's more Darwinian if you think about it. That's actually how Jennings likes to describe it. So Paramecium is sensitive to lots of different chemical substances. It's also sensitive to temperature, to light, so it's photophobic mainly, to water currents. It also shows social behavior through sensitivity to carbon dioxide, which it produces when it breathes. And there might be other things as well, because it's known that they have GABA receptors and they release GABA as well. And all of these are mediated by ionic channels that produce transduction currents. So briefly, I will pass briefly in other types of behavior that you can find in the Paramecium. One is jumping. So here you can see a slow down movie from a recent paper where they use strong heat stimulus, which is a laser, and then you can see Paramecium jumping. It jumps by throwing little spears called trichocysts, and these are little spears docked near the membrane that it throws. It's calcium-based. It can also bend in narrow channels because it's flexible. So for people interested in embodiment, it's a really interesting model organism. And here can see is the escape reaction. When you touch the rear of Paramecium, it speeds up, moves forward a little bit. And that happens also when you tap on a slide, which I did here during the lockdown. All right. So in addition to that, it also shows adaptation. So I briefly talked about adaptation on a fast time scale to the chemical environment. It also adapts on a long time scale of a couple of hours. And that's important because a neuron has as extracellular medium something that is controlled normally. But for Paramecium, which is a single cell, the extracellular medium is the outside world. So it's very fluctuating. The composition, the temperature can change. And so the ionic channel, all the metabolism, etc., has to still work robustly when the environment changes. And so here what you see is the adaptation of velocity, so one aspect of behavior with time when you change Paramecium from the culture medium to some other ionic medium. And so after a couple of hours, you see that the behavior adapts and becomes as it was before. So there is some homeostasis of behavior through changes, actually, of physiological properties of channels. There's also adaptation in temperature. That is, there is thermotaxis, where Paramecium swims towards the temperature at which it has been adapted. And finally, there's also some literature on learning. What you see here is one example which I find convincing. There are actually quite a number of papers on learning in Paramecium, but they are controversial because it's a bit difficult to show clearly. So here what they did is to condition Paramecium to respond to sound. And so normally you have trajectories that look like that, or in spiral trajectories. So when there's no stimulus, and in B, you can see what happens is you present 500 Hertz tone, nothing. But if you pair it with an electrical shock in the middle of the presentation of the tone, so 2 seconds after that, that triggers an avoiding reaction, so it changes direction. And if you repeat that a few times, then Paramecium starts the avoiding reaction before the electrical shock, just when you present the sound. So it has learned to respond to sound. Okay, now I will show some ongoing work that we are doing on integrative modeling of the avoiding reaction, so one aspect of the behavior of Paramecium. Integrative in the sense that we're trying to look for an electrophysiological model, Huxley type model, but also with the coupling with motricity. And so in detail, what happens during the avoiding reaction is that, so the cilia revert, the cell swims backward, then it turns and it swims forward again. So how does it turn actually? It turns, probably, it's not entirely clear, but it seems that it turns because the cilia in the rear and the front revert asynchronously. And so it turns around the mouth. Very briefly, I wanted to mention some work that my collaborators are doing on analyzing behavior and microengineered swimming pools with obstacles and tracking. So I won't discuss that today. I will talk about the electrophysiology mostly. So the main difficulty for the electrophysiology is that it swims really fast. It swims at about 10 body lengths per second. So the first thing we need to do is to immobilize it. And in the 70s, they used a fairly complicated technique, which I won't discuss. But because it was a bit technical, we tried to develop something simpler, which is based on aspiration. So we put a filter that has holes that are smaller than the cell, and then we pull the fluid from below with a peristaltic pump, and then the fluid goes back. And so you can see here, I'm not sure you can see the movie clearly, but the cell is immobilized against the filter, and you can see the cilia beating. And once you have that, you can stick 2 electrodes in it, stop the pump, and then the cell is kind of pinned to the filter, to the slide, and then you can do some electrophysiology. So here I'm just showing some, maybe not the most interesting part of electrophysiology, but it's to explain briefly what kind of currents there are in paramecium. So these are just responses to long pulses. And in response to long pulses, you have graded action potentials, which are basically calcium and potassium based. There's a transient calcium current, which you can kind of guess as an inflection here. It inactivates in a millisecond or 2 milliseconds. It inactivates not with voltage, but with calcium itself. So when calcium enters, it binds through calmodulin and inactivates the channel. This type of calcium channel actually was discovered first in paramecium and later in neurons. So there's this calcium channel, which is located in the cilia, and then there's a classical delayed rectifier potassium channel, which is not in the cilia but in the basal membrane. And while there's calcium in the cilia, the cilia revert and beats in reverse. In the hyperpolarized region, there are also some interesting things going on, but I won't have time to talk about it much. Basically, it controls the escape reaction, which is what happens when you touch the rear of paramecium. And the cell speeds up, it contracts, things like that. Okay, now we're trying to basically build a Hodgkin-Huxley type model of the electrophysiology. So what we're doing is, instead of using pharmacological manipulations, we use some other type of isolation, which is mechanical isolation. So the first thing we do is to remove the cilia. In fact, the calcium channels are in the cilia. So once you remove them, you remove the calcium current. You also remove calcium-activated potassium currents. I didn't discuss them actually, but there are also calcium-activated potassium currents. And so what you get is just the potassium current. So the way you do that is you put the cells in alcohol, you shake, and then the cilia fall down. And if you do that, you get a nice cell without cilia, like round and nice, and the cell is alive. In fact, within a few hours, the cilia grow back and it can swim again. So it's still a recording, in vivo awake recording, okay? And what you see here is just responses to pulses. And then you can use some model fitting techniques. We use that instead of voltage clamp because controlling the voltage was actually not so easy with high resistance electrodes. So we just use current clamp and model fitting, just on the depolarized part, because that's where the avoiding reaction happens. So, so we inject a random current, and then it's actually quite easy to fit a delayed rectifier model to this data, except we have to take into account that the channel inactivates apparently. So it inactivates with voltage. This is actually quite classical with delayed rectifier currents. So that's fairly easy. The more difficult part is the part that deals with the calcium, because we don't measure the calcium directly, at least not for now, and so we have to infer it. So there is some literature, actually there's a lot of literature on electrophysiology in paramecium. It's just that there is no model. So we know that there is an inactivating calcium current. So you see an example here in voltage clamp. And it activates with calcium. So you can model that with a Hill function, basically. Then you need to model calcium dynamics. So calcium dynamics are modeled by, well, calcium that enters, which is due to the calcium current, and the calcium that goes out with pumping. There are different ways of modeling this, of course. And there's also calcium-activated potassium current. This you can show by injecting long pulses, and then you can see a hyperpolarization at the end of the pulse. There's actually a little bit of literature, some studies in patch clamp. All I showed before was microelectrode sharp recordings, but some people, actually 2 people, have done patch clamp recordings in blisters of paramecium. So you put some chemical that makes blisters on paramecium, and then you patch. And that's fairly difficult to do, but there are some single channel recordings. So from the literature, you can kind of guess some parameters for these models. And so with the hand-tuned model, you get something like this. This shows responses, graded action potentials, responses to current pulses. So you can see the delayed rectifying current, inactivating calcium current, and then the calcium concentration that increases with that current, and then it's pumped out. And what you can see is that the duration of this increase in calcium depends on the stimulus. It's graded, and that implies that the backward swimming would last longer with greater stimuli, and this is something that is known also in Paramecium. So you can get some qualitative results in this way. To fit the model is a little bit more difficult because we have 3 different currents and calcium dynamics together. So this is just some work in progress, but it's not finished. Now what I want to show is the next step, which is the coupling with the motricity. And so what we do is the following. We put some particles, silica particles, in the medium, and then when the cilia beat, the particles move, and we can monitor how the cilia beat in this way. Actually, this is a little movie. I'm not sure it's very clear on the left, but you might guess some particles moving. And on the right, it's an analysis called particle image velocimetry, which shows the vector field, so the movement of the fluid. And I play it again, sorry. I can't play it again. Can I play it again? You can see the vector field switching back and forth, and that's what happens during the action potential. Then this is just some preliminary analysis, which shows that the angle of this vector field changes in an all-or-none way with the stimulus amplitude, but the duration of the backward swimming basically is graded. So we have ways to measure that, and then the next step is to couple that with the Hodgkin-Huxley model, and specifically with the calcium variable in that model. And finally, and this we haven't started yet, but there is some literature on it, we want to couple the model with models of transduction currents, in particular mechanical transduction. So what is known is that when you touch the front of the cell here with a glass stylus, you get a short-lived current, a depolarizing current, which is mediated by calcium. And if you touch on the rear, you get a potassium current. So you have a gradient of mechanosensitivity from rear to bottom. And actually in the genome, which has been fully sequenced, there are piezo genes, which are mechanosensitive, genes for mechanosensitive channels. So, and I finished on this thank you slide. So these are my collaborators, Alexis Prévost and Laetitia Pontani, my physicist collaborators from the Laboratoire Jean Perrin. They're working mostly on behavior, trying to do in particular analysis of 3D trajectories. And Anirudh Kulkarni did some of the electrophysiology as a postdoc. Nicolas Escoubet is a PhD student working on stereo, so 3D recordings of trajectories. Manuel Beran did the first electrophysiology recordings in the lab during his rotation. Mette Demir-Sigil did the first behavioral experiments. Irene Elises is also doing the electrophysiology and the model fitting now. And finally, Eric Meyer is a geneticist, expert of Paramecium genetics in ENS, with whom we're working to use genetics tools. There are really interesting genetic tools in Paramecium, such as RNA interference, and also, well, the genome has been sequenced, and it's possible to develop calcium imaging sensors. That's what we are trying to do now. So thank you for your attention.
Okay, thank you for your talk. It was very nice. Regarding the questions, you can either post them in the chat, or you can demand to or request to join us via video, and we can add you here to the video chat so you can ask Romain directly. Until then, I wanted to ask, so you showed this first a literature question, basically this pictures where the Paramecium learned to be shocked by, to stop swimming by sound. So do you have any idea how the mechanism behind that works, or?
So the paper doesn't talk about mechanism, so it's purely behavioral. So it's not known. But what we know is that, so the avoiding reaction is calcium-based. So there's an influx of calcium in the cilia when you do the electrical shock. And then, well, the sound is a mechanical vibration, and so that probably produces a transduction current, which might be calcium-based as well. If it produces an avoiding reaction, then probably that means it depolarizes the cell, so it's probably calcium-based. So there's probably some calcium pathways there, and then I don't know.
Okay.
I would put a question to you from Christine Tesma-Reible, our next speaker. And she's asking how similar or heterogeneous the electrophysiological responses are if you measure from like several different parameters.
So do you mean different individuals of the same species or different species of Paramecium?
I guess both.
Yeah. Both.
Okay. Yeah, I can answer both. Sure. Well, so the first big difference, but it's not very interesting, is that they have different sizes. Different species have different sizes, and also during development they change size because so they divide, and so basically the size is divided by 2 for a little while, and then it grows back. So and that changes the magnitude of the currents. Okay, so that's an aspect that is kind of boring. Then if you look at individual differences, we see important individual differences. In the literature, it's not so clear, but I think some of the currents, I think it depends on the channels actually, but some of the channels can vary quite a bit. And presumably there are differences which are not genetic, because actually genetically they're all almost identical, all the ones we use. Not genetic, but depending on the metabolic state. Because all these channels, and it's in fact not specific to Paramecium, it's just that in Paramecium it's more obvious maybe. All these channels, they can be modulated by various things like cyclic nucleotides and things like that. And for example, what is interesting in Paramecium is that you can see how that relates to behavior. Because I didn't mention, but apart from bouncing around, what it does is feed. And to feed, it attaches to a fiber like decaying plant or things like that. It stays there, and the oral cilia keeps beating and pull some bacteria, because it eats bacteria, into the mouth. And while it's doing that, well, it's not swimming. So some of the cilia stop beating, and things, and different things happen. and it's less reactive mechanically. So it's basically in a different metabolic state, and that certainly implies changes, physiological changes to the ionic channels. And but it hasn't been shown clearly because there is no preparation for the feeding behavior that I've seen in the literature. It's something also we're trying to do. But yeah, so there's certainly variation, which I think is due to metabolic changes. I forgot to mention there's a circadian rhythm in Paramecium and an ultra-circadian rhythm as well. So it's an organism.
Yeah, there's one more question by Wilhelm Braun, also one of our speakers. And
okay.
if you could elaborate a bit more on the crowd effect. So you mentioned that they can...
Yes, so absolutely. So and this has been known since the 19th century. They tend to gather in different places. So, for example, if you put them in the depression slide, after a while you will find them gathering in the middle at the bottom. Or if you put them in your tube, they will they could gather at kind of random positions in your tube. And what happens? So, according to Jennings, who described that in his book, it's a combination of breathing and chemotaxis. So when it breathes, it produces carbon dioxide, and it is attracted to carbon dioxide. And therefore, it just by this, it tends to gather. So feeding, for example, uses a combination of, well, not sure exactly what happens, but probably some kind of mechanical sensitivity to attach to somewhere, a fiber, but then there is production of carbon dioxide, which kind of attracts other specimens, and then suddenly you have lots of Paramecium feeding around the plant. So GABA receptors, yeah, I mentioned that. So there are a couple of papers. One or two show that there are GABA, I think A and B actually, receptors in Paramecium, and other papers show that they produce GABA. They release GABA, but apart from that, I mean, it's a little bit anecdotal in the sense that I don't know in what conditions they produce GABA, and I don't know if it is the basis of some kind of social behavior. It's not known. At least there is something, that's for sure. Yeah. Temperature, yes, very interesting. Okay, yes.
I think there's someone else before me. I don't know. I don't want to intrude whether Leonidas wants to ask first. But
is there?
Fabian, you decide.
No, he answered this. He just answered Leonidas' question. He jumped right ahead.
Oh, sorry. Then okay. Then I missed that. Sorry. So I've got my temperature question, which was very obvious. We discussed it before, but it's a wonderful organism to study how the behavior changes with temperature. So can you remind me, how stable is it across what range of temperature? And I'm sure there must be some differences in terms of reaction times or some qualitative differences in terms of behavior. And also relating to Christine's question with the circadian rhythm, we know that in mammals also temperature can entrain the circadian rhythm. Anything known about that for Paramecium?
Okay, so first the circadian rhythm. So there are a few papers on that, but I think they only use light as to entrain the rhythm. So I don't know if it has been done with temperature. I would think not. On temperature, there are quite a number of papers actually on that. It's very interesting. Some from Jennings, or old paper from the 19th century, which shows so that Paramecium can survive between close to 0 degree and maybe 40 degrees, something like that. And then when it tends to prefer, so to gather at the temperature that is close to the temperature at which it was cultured, or just adapted, in fact, or after 3 hours, something like that, it adapts to the new temperature. And then there's also electrophysiological work by Mashmer. I can send you the references if you're interested. just remind me. And there's some very interesting work by Meshmer showing what happens with the unit channels when it adapts to temperature. And so indeed, the conductance of the channels change and so on. Of some of the channels, like the calcium channel and the potassium channel. And the behavior itself is mediated by temperature-sensitive channels of different types, which are some sensitive to cold and others to heat, basically. So yeah, there must be a dozen papers on that, something like that.
Thank you very much.
Okay, I would say we stick to Kai Kasper's question as for last, and then we get Christine on stage.
Okay. Yes. Yeah. So I'm not sure it's especially complex, actually. If you look at the microbial world, it's just amazing. It's just that we're not used to it. So Paramecium just happens to be the model organism for ciliates because it's very easy to find and it's big. But there are lots of other ciliates which do amazing things. There's, for example, Lacrimaria olor. I don't know if you heard about it. It's a ciliate which has a flexible, extensible neck, which is kind of spreads and throws to explore its environment and eat stuff. I mean, there's a whole zoology there. About learning of classical conditioning, I'm not sure. I think there is some work which I don't know very well on stentor. Stentor is another ciliate which has other types of behavior. It tends to be attached, and then if you touch it, it retracts and things like that. So there's a little bit of work in stentor and probably a few others as well. Yeah, and the calcium channels in the cilia, they are are they sensors as well as effectors? They're not sensors. Right.
They wouldn't be opened or closed if you artificially move the cilium.
Well, so from what I've read, for the mechanical sensitivity, it's based in the basilar membrane. But when you move the cilia, of course, you have a mechanical signal that spreads along the cilia. So indirectly, it brings some sensitivity and some complexity to the mechanical sensitivity. But the mechanosensitive channels are in the base and not in the cilia. At least that's what is known. Hydrodynamical coupling. There is hydrodynamical coupling too, for sure. Yeah, in beating. Beating is coordinated by hydrodynamic coupling. Actually, there are waves of beating on the surface.
Okay, I think we have to move on.